Okay, all right. So, hello, dears. Good day. All right. So, welcome to yet another pre-recorded lecture in our bacteriology lab class. And for today, we're gonna start now with biochemical testing part 2, which I believe is another TMI na gyud, if not the most TMI na na mga na part. Okay. All right. So, yeah, for this, again, just try your best to internalize it at your own time, at your own pace, because again, it's too much to take in at the first glance pag yun, or first study. So again, take your time. Kaya nyo yan. Alright, okay. So again, this is biochemical testing. As mentioned, di ba, nahuman ito sa part 1, nag-gloom rata atong Haemophilus and Neisseria, di ba, gram-neg-cocci. Because again, of mga certain complications of the COVID stuff diri sa Duma. But now, again, I'm back at this type of lecture, pre-recorded lecture. And this time, what we're going to talk about is, again, biochemical testing part 2. We'll start first with the gram-positive bacilli. Okay, all right. So, the gram-positive rods or bacilli. Okay, all right. So, what we're focusing here usually is aerobic gram-positive bacilli. Because, again, there are gram-positive bacilli that are anaerobic. Sino yon? Of course, Clostridium. So, again, anaerobes, parehas ni Veillonella sa cocci, gram-negative cocci. They are discussed as one, okay, regardless of their gram-stain reaction and morphology. Munang wala na ako gilabot sa ang Clostridium din because again, it would take time po kay ubay-ubay po ang test ni Clostridium. But for now, what we're going to discuss again are your aerobic gram-positive bacilli. Most species that are found in the environment and can be isolated daw in the soil and water. Most, again, are not highly pathogenic, but their isolation in the clinical lab are increasing in frequency. Okay, all right. And divided into groups, this is according to Mahon. Okay, all right. First is the non-spore forming, of course. Wala sila nag-release of spores. Non-branching, meaning ilahang morphology wala ga branch. Like, wala sila ga branch na murag filaments or murag fungi. Okay, so that's Corynebacteria, the Corynebacterium species, and Rothia spp. We have the non-spore-forming, non-branching, but catalase negative. You have Erysipelotrix rhizopathy, Arcanobacterium, Truperella, and Gardnerella vaginalis. You also have the non-spore-forming, pero branching this time, actinomycetes. Okay, so these are again the fungus-like bacteria because again, their appearance under gram stain was like fungi, but they are not really fungi, they are bacteria. Okay, so the actinomycetes, nocardia, and other actinomycetes. And the last type is, of course, the spore-forming, catalase positive, pero non-branching, the Bacillus spp. Okay, di ba? Remember, ikapila na ako na ibalik-balik, ikapila na ibalik-balik. Kinsa tong two medically important genus of bacteria that produce spores, you only have Bacillus and Clostridium. And Clostridium, remember, is again anaerobic, meaning they don't need oxygen or they don't want oxygen. Whereas for Bacillus, they need oxygen or they produce spores in the presence of oxygen, di ba? Okay, so again, they are aerobic. So appeal sila ani na discussion. Ang Bacillus. Okay, so here's an example of pictures. This is, again, nocardia. As you can see, branching. Filamentous. More like mga filaments. Okay? Alright. Next one, this is, of course, na yung mga spores sa tunga, na yung mga kanang murag halo sa tunga or murag oval sa tunga. Those are your spores. And this is Bacillus. Yeah, spores at the center. So, central na spore. And you have, this is Corynebacterium diphtheriae. Remember, for Corynebacterium diphtheriae, again, Chinese letters, diba? Chinese letter appearance, yes, na mga, or Chinese characters. Basta gani ka na under gram staining or case study man gani mo ingon na Chinese characters, press the buzzer, that's Corynebacterium diphtheriae or Corynebacteria. Alright? Okay. And unsa ganito ang metachromatic granules ni C. diphtheriae, katong na-mention na po na ako sa intro to micro, we have inyong batchmate si Babes Ernst. Diba? Si Babes Ernst granules. Okay. That's really specific for C. diphtheriae. Okay. Alright. So next we go now to their biochemical testing. So what tests should we perform to identify them? So here's an example of a flow chart on how to identify. Okay. So this is really short lang yun. So if you see under the gram stain, nakakita ka gram-positive rod. Okay. Next, you check if it's a spore former or non-spore former. And then you perform the other tests there. Okay. So we'll try to discuss them individually. Okay? Alright. And mga other inputs put. Okay. So for biochemical test for identification, we have again catalase test. Again, na-mention na nato, di ba? Catalase test. Detects the enzyme of catalase. Positive reaction nato is effervescence or bubbling. Second is hemolysis on BAP. That's usually used for the Bacillus species between B. anthracis, Bacillus anthracis, and other Bacillus species. Okay? Test similar to Enterobacteriaceae, which is ang second part nato sa or actually the first part dapat di ay supposed to be. Okay, but again, I'll recommend to start viewing first the Enterobacteriaceae before this. But again, gilate rin ako siya record kay taas ang Enterobacteriaceae. Medyo daghan ka ayong stuff to remember. Okay? Alright. So, again, their tests are quite similar to Enterobacteriaceae, like H2S production, TSI, triple sugar iron, urease, and nitrate reduction. And we have the specific tests or characteristics for each species na itong i-look into today. Okay, so we'll start first with the first test. For this table pala. So, these are example of a summarized na gyud of the tabular summary of some of the aerobic gram-positive bacilli. So, I'll just point out mga important stuff. Okay, alright. Listeria monocytogenes, guys, please take note, it's esculin hydrolysis positive. Okay. Listeria monocytogenes, if you can recall sa ato ang biochemical testing for Streptococcus, it's also CAMP positive, remember? Okay, CAMP positive together with S. agalactiae, diba? So, usually, pwede silang ma-confuse, but you don't have to confuse them because, so if mo ask mo, sir, if I have CAMP positive reaction, so it could be Listeria or Streptococcus. So, how do I differentiate them? Wag di mag-uol. Easy ra. Gram stain palang daan. Listeria is Bacillus and Streptococcus agalactiae is a cocci. So, dito palang daan, ma-differentiate na niyo ang duha. Okay? So, kung Bacillus siya niya, CAMP positive, then that could be Listeria monocytogenes. Okay? Another one is, of course, it's esculin hydrolysis positive. Motility. Ang saglit characteristic na motility sa Listeria, the tumbling motility, di ba? Tumbling or end over end. Ayan, end over end. Motility. Tumbling, motility, end over end motility. Ikapila na sige balik-balik, right? So again, that's Listeria monocytogenes. Okay, alright. Ayan. Okay, next. Erysipelotrix rhizopadi, my favorite, because nice kaya ngalan. And very, very characteristic yun niya is, guys, as you can see, siya ray mo produce og H2S in the TSI production. Again, H2S production, mga inana, that would be explained sa Enterobacteriaceae, kay daghan po kayong H2S producers there. But please take note, gram-positive bacillus, okay? Gram-positive bacillus, good. And then, nakakita kang H2S production, press the buzzer. That could really be Erysipelotrix rhizopadi, okay? There are some bacillus daw na ma-produce og H2S, but it's not that kanang common or dili kayo siya ma-detect. But Erysipelotrix rhizopadi, basta gani gram-positive bacillus, mo produce og H2S, as in first organism that could come to your mind is Erysipelotrix rhizopadi. Okay? But again, kung gram-neg bacillus pero H2S producer, daghang pwede. Okay? But for gram-positive na bacillus pero H2S production or H2S producer, then that could really be Erysipelotrix rhizopadi. As in, very, very characteristic yun na niya, guys. Wag kalibutan, Erysipelotrix rhizopadi. Okay, unsa pa man. Alright. I think that's for now. Okay, ayan. Alright. Okay, again, that's a characteristic lang of some of our aerobic gram-positive bacilli. Okay, so first we'll go now to the production of halo on CTBA. So, ang purpose is we detect the presence of the enzyme cystinase. Okay, and the culture medium used is cysteine tellurite blood agar or CTBA. And the principle is again, we inoculate organism on CTBA, incubate at 35 degrees Celsius under capnophilic requirements, 5 to 10% CO2, and we observe for black colonies with surrounding brown halo. Here's an example. Alright, black colonies with brown halo. And we usually use this for the identification of the Corynebacterium diphtheriae and other species. Okay, so CTBA, production of halo on CTBA. Okay, again, detects the enzyme of cystinase. And ang positive is na ay black colonies per na brown halo. Ayan, as you can see. Which detects the presence of detects the presence of the enzyme cystinase because ang cystinase i-break down ang cysteine, okay, and then mo react with the tellurite munang na ay brown na halo, okay? All right. Ayan. Okay. Next we have the gelatin hydrolysis test. When you see gelatin hydrolysis test, hydrolyze. Okay, so we detect the presence of a gelatinase, so mga enzymes pa rin, that could liquefy gelatin. Okay, so again, we inoculate organism into a gelatin medium, which is a deep, okay, or a butt. And then we incubate for up to 14 days. And then we remove daily and place at 4 degrees Celsius to check for liquefaction, or meaning if gahi ba siya ni liquefy, meaning nahimo ng liquid. Okay? And we observe for liquefaction if the control na atong gigama pod is ni harden na, meaning we have a control, okay, which is uninoculated na gelatin medium, and then if nigahi na siya, okay, dito ta mo confirm sa liquefaction sa katong ato ang na-inoculum or na-inoculate na tube. Okay. So here's an example. Of course, letter A mo na siya positive, liquefaction, meaning ni liquid na siya. Okay? And for letter B, remember, di ba, nigahi ra siya, walay murag liquid. So that's negative. Or, this is the control. Okay? Okay? Alright. We use this again still for Corynebacterium and other also bacteria. Bacteria. Dili ra for gram-positive bacilli, napod sa gram-neg. Okay? Alright. And we have the ELEC test. This is really specific food for C. diphtheriae because it detects the presence of the diphtheria toxin. And it's an in vitro, meaning outside of the, dili siya kanang, it does not use, or it's not performed inside the body. So in vitro, like in the lab yun. Di ba, in vivo testing for diphtheria toxin is, it uses, yes, it uses guinea pig, di ba, remember? The guinea pig lethality test. You use the guinea pig, so in vivo na siya, meaning you use it, you inject it inside a living body. Okay? Alright, ayan. Ang principle is again, immunodiffusion. So as you can see, we streak the organism parallel to each other. Okay? And then, 10 meters apart. And then after, we put a strip na naa dito ang anti-diphtheria antitoxin. Or like, it's quite, in a nutshell, or parang in a rough term, it's an antibody against a diphtheria toxin. So, diphtheria antitoxin. Okay? And if ever manggani, ang organism ni release of diphtheria toxin, mo react siya with the antitoxin, they will create precipitin lines. Okay? Lines of precipitation. So, here as you can see, this is the control. Okay? Ayan. Or in a kanang lab setting yun. This is the control. Okay? So, meaning, ang control mismo, nag-release yun siya what they call this? Toxin. Okay, control. Known organism siya to release toxin. Okay, so pag butang sa strip, of course, na ay precipitin, meaning nag-react ang toxin na diphtheria and the antitoxin found in the strip, munang ninay precipitin lines na na-create. Now, for number 2, example is the unknown organism. Okay, so therefore, ni-release man siya og precipitin lines. What happens? Ang precipitin lines sa 2 og 1 mag-connect. Okay, and that is what we call the arc of identity. So meaning, they are of the same arc of identity. Okay? Arc of identity, meaning they are of the same toxin. Because ni combine man sila, it means they are of the same substance that creates the toxin. Okay? Alright. Okay, ayan. Alright, and negative control, number 3, of course, walay toxin, so walay precipitin lines na itabo. And, okay, number 4 is the another unknown organism. If you want to detect if ni-release ba siya toxin. And this time, wala man. So therefore, it's negative for the toxin. Okay? Gets? Combine sila. This is again the arc of identity, which means similar ilahang toxin kay ni-combine man. Okay? So they are of the same na reaction. Munang ni-combine. Okay? That's the arc of identity. That's why we call it arc of identity because in a way, you're confirming the identity of the second ni unknown. Diba? Unknown organism. You're confirming its identity na same yun siya sa positive control, which is known already na mo-release siya ani na toxin. Okay? In this case, the diphtheria toxin. Okay? Alright, that's the ELEC test. Usually used for C. diphtheriae and again to detect if you have the diphtheria toxin. Okay, alright. And summary results lang of the Corynebacteria. So as you can see, for the halo and CTBA, guys, ang common na yun, kanang tulo na yun na mo-produce ana is C. diphtheriae, ulcerans, and pseudotuberculosis. Now, sir, how do we differentiate? So, if you really want to isolate C. diphtheriae, yan ay CTBA halo, you use urease. As you can see, siya rin negative sa tulo. Right? Positive ang ulcerans and pseudo-TB, but ang diphtheria is negative. Really, very characteristic po sa C. diphtheriae, it's urease negative. Okay? Compared naman sa other tulo. Okay, alright. Next, for gelatin hydrolysis, kinsa rin positive? C. ulcerans. And for nitrate reduction, ubay-ubay ang positive, si diphtheriae pa rin and pseudo-diphthericum. Okay. Alright. For escalene hydrolysis, negative sila tanan. Okay. Alright. So again, very characteristic ni C. diphtheriae, it's urease negative. Okay. Alright. Especially if you want to confirm it or you want to compare it against the other two species na na-halo on CTBA, si ulcerans and pseudotuberculosis. Okay. Again, urease. Perform the urease test. Negative si diphtheriae, positive si ulcerans and pseudotuberculosis. Okay. Alright. Ayan. So that's for the corioning bacteria. Okay. Next is test for motility. Okay. So again, na-mention din sa Enterobacteriaceae, which is again, mas in-depth to dito pag explain. Okay. Alright. So again, it determines whether the organism is motile or non-motile. So ang principle is again, flagellated organisms, they move away from the stab line or from the point of inoculation. So here's an example, di ba? The left is motile, the right is non-motile. Very common na sa atong hanging drop test for motility. I'm sure you're aware of that already. And here, again, ato lang i-mention mga characteristic na motility of some organisms in a semi-solid medium. Okay, so example, si Listeria monocytogenes, because of its tumbling motility, ang iyahang appearance in the semi-solid medium is described as umbrella pattern. At room temperature room temperature. Kaya mawala siya, dili siya mo appear if like 37 or 35 degrees Celsius. So dapat siya at room temperature, okay? Umbrella pattern, okay? Of in semi-solid. For next is you have Erysipelotrix test tube brush. As you can see, more siya test tube brush. But Erysipelotrix is non-motile, guys, ha? Inanira gyud iyang growth sa semi-solid. I don't know why. But again, it's considered as non-motile, pero iyang growth is described as test tube brush. As you can see, diba, siya test tube brush. Okay. Alright. Still the same at 22 degrees Celsius or at room temperature. And lastly, Bacillus anthracis, this one. Again, Bacillus anthracis is non-motile. Okay. Most of the strains are non-motile, but there are some strains down na motile. And if they are inoculated daw in a semi-solid medium, it's described as inverted pine tree appearance. Bacillus anthracis. Okay? Diba? As you can see. Motile strains, inverted pine tree or inverted fir. F-I-R, fir, inverted fir or basta pine tree appearance, okay? For Bacillus anthracis. But generally, guys, Bacillus anthracis is non-motile yun. Okay? Alright. Ayan. So that's for test for motility. Okay. Next, we go now to the cold enrichment technique. Basta ganun for cold enrichment technique, guys, ang very, di ba, na-mention na po na ako sa intro to micro, if wala ko nasa yupjod. Cold enrichment technique that's usually used for Listeria monocytogenes, especially if polymicrobial, meaning daghan ka ayong kagaw na naa sa specimen. Okay, so if you really want to isolate Listeria monocytogenes, you can perform cold enrichment technique. So how do you do this? Yeah, the growth lang siya at 4 degrees Celsius. Because again, Listeria daw has a wide range of temperature growth. Pwede as low as 0.5 to as high as 45. Okay? So what you do is the specimen is placed into a broth, okay? And then you incubate for 4 degrees for several weeks. You subculture every week, and then we examine for L. monocytogenes. It's either we perform gram staining, example, L. monocytogenes po niyang result, or we inoculate it into BAP. Sa BAP, it is resembles the growth of S. agalactiae. Okay. Or again, you can always perform motility, umbrella pattern, other tests, escalene hydrolysis ba, okay, or CAMP test. Okay, di ba? Ang CAMP test sa Listeria monocytogenes, it's block shape. Okay, di ba? For S. agalactiae, arrowhead, right? But for Listeria monocytogenes, it's block. Okay, block. Like, block. Okay, block shape of hemolysis for Listeria monocytogenes sa CAMP test. Okay, alright. Ayan. Next, if we have the CAMP test, we also have the reverse CAMP. Now, say points sa reverse CAMP. Okay, alright. Now, ang purpose niya is we determine if na by synergistic relationship between two microbes. And specifically for gram-positive bacilli, we used it for Arcanobacterium hemolyticum because A. hemolyticum releases phospholipase D, which inhibits the beta-lysine produced by the S aureus. So, what happens is it is a CAMP inhibition reaction. So, muna siyang, it's called reverse CAMP. Kaya in a way, di ba, ang CAMP test, recall in CAMP test, nahitabo ang CAMP test because iyang gi-enhance ang hemolysis, remember? Because ang S-agalacti mo release, agalacti mo release siya og camp factor which enhances the hemolysis produced by S. aureus, di ba? Munang na arrowhead hemolysis. But for this type, reverse camp, na-inhibit noon siya because Arcanobacterium hemolyticum releases phospholipase D, which inhibits the beta lysine produced by S. aureus. Muna siya in a way, reverse camp siya. Okay? Alright. So again, strict na BAP ang hemolyticum, strict perpendicular ang S. aureus, incubated, and observed for arrowhead hemolysis pa rin daw. I'm not sure if, again, wala man good mention sa Bailey and sa Mayhon kung say positive reactions sa reverse camp. But of course, as you can see sa picture, pwede arrowhead, di ba? As you can see, arrowhead ragyapon. Right? So, so ang positive again is A hemolyticum. As you can see, di ba? Dirigi streak ang A hemolyticum. But pag abot niya sa Staph aureus, naay murag hemolysis or murag inhibition, naanay ni inhibit ang growth, wala na siya ni tubo dito. Instead of tubo, murag naanay hemolysis na pag arrowhead. So muna siya point ana. As you can see for iyahang counterpart, T pyogenes na, Truperella pyogenes na ni siya. Nipadayon o grow until sa streak, 'di ba? So meaning that's negative, 'di ba? Streak intersected the S. aureus. Nipadayon siya. Because again, wala siya phospholipase D. Okay, all right. Now for reverse camp test, this is also found usually used for C. perfringens or Clostridium perfringens. Okay? Clostridium. Now, for reverse camp sa Clostridium manggod, ang ginagamit na sa reverse camp sa Clostridium is, instead of Staph aureus ang streak, Strep agalacti ang imong streak sa tunga. So, I think muna siya pasabot sa reverse camp for C. perfringens. Kay, instead of Strep agalacti ang imuhang test organism, siya na noon ang reagent. Siya na imong streak sa tunga. Okay? Alright. That's for Clostridium perfringens. But for A. hemolyticum, again, remember, phospholipase D inhibits the beta lysine producing S. aureus. So what happens is again na inhibition. So as you can see, parang arrowhead ragya po na iyang appearance, no? But again, according to Mayhon and Bailey, so wala gyud gi-mention kung say positive reaction, ha? Okay. Alright, but according to picture again, basta take note lang na wala na ni padayon o grow si Arcanobacterium sa streak sa S. aureus. Di parehas ni Truperella pyogenes na ni padayon ragya po siya. Okay. Alright, because again, the presence of phospholipase D. Okay, all right. So for the next part of the video, we'll now go to the remaining tests na for your gram-positive bacilli. Okay?